Hey everyone, welcome to this explainer. I'm absolutely thrilled to have you here today. If you've ever stared at the output of a chemical analysis, those fascinating squiggly lines climbing up and down a graph, and wondered how on earth scientists pull hard, precise numbers out of them, you are in the exact right place. Today, we're decoding the mystery of quantitative chromatography. We're going to explore exactly how we figure out the precise chemical quantities hidden inside those peaks and valleys. All right, let's dive right in. So here's the fundamental mystery we're tackling today. Chromatography is incredibly famous for its ability to separate a really complex mixture into its individual components, right? But that's just the qualitative part. How do we actually bridge the gap between simply seeing a separation on a screen and measuring a physical mass? How do we point to a peak on a graph and say, with absolute certainty, there are exactly 5 nanograms of this specific compound in this sample? That is the puzzle we're solving today. Here's our roadmap for the day. We'll hit the power of chromatography, dive into the core linear equation, and look at the measurement problem. Then we'll break down 3 methods to solve it: external standard, internal standard, and normalization. Okay, section 1, the power of chromatography. When we're talking about this kind of quantitative analysis, we are looking at a globally trusted technique. We're talking about the strict EPA methods used for trace and ultra-trace environmental analyses, the exact stuff keeping our air, water, and food safe. And today, modern tech really does the heavy lifting for us. Instead of the old-school days of manual triangulation, a microcomputer just swoops in, digitizes the signal, and converts the physical area under those eluting peaks into our raw data. The software completely automates the calculations for retention times and peak areas. But having the peak area, that's really just half the battle. We still have to figure out how to convert that visual area into an actual mass. Moving on to section 2, the core linear equation. To actually pull a concentration from a peak in our chromatogram, we rely on one fundamental mathematical relationship. And here it is: mx equals kx times ax. Let's break that down real quick. The mx is simply the injected mass of our compound. Ax is the area of that eluting peak the computer just gave us. The assumption here is that there's a strict linear relationship between the injected mass and the peak area. But to actually make them equal, we need a multiplier. Think of it like a mathematical decoder ring. And that is our kx. So what exactly is kx? Well, it's known as the absolute response factor. It's the variable that connects the area of the peak to the actual quantity of the compound. But here is the massive catch, and it's a big one. Kx is not some intrinsic property of the chemical itself. You can't just flip open a textbook and look it up. No, it depends entirely on how the chromatograph is tuned at that exact moment. Literally, if you tweak a single setting on your detector, your Kx completely changes. Which leads us directly into section 3, the measurement problem. Okay, so we've got our basic equation, right? But as any of us who've spent time in a lab know, the physical world is messy. We absolutely cannot just trust the math blindly because we run into 2 huge hurdles. First up, to even calculate our Kx decoder ring, we have to know the exact injected quantity. But getting a perfectly precise injected volume, it is nearly impossible. We're dealing with microscopic amounts here, and there are strict mechanical limits to the precision of syringes and injection loops. Second, like we just talked about, Kx is constantly shifting with instrument tuning. Instrument drift is a very real thing. So if our injected volume is always slightly off and our decoder ring keeps changing, how in the world do we get an accurate measurement? That brings us to our solutions, starting with section 4, method 1, the external standard. This is where human ingenuity really kicks in. The external standard method is a brilliant workaround for these physical limits. It's a pretty straightforward step-by-step process. Step 1, you inject a specific volume of a reference standard with a known concentration. Step 2, you measure that reference peak area. Then, and this is absolutely crucial, without changing a single condition, you inject the exact identical volume of your unknown sample. Because those injected volumes are identical, we can just completely ignore K sub x. We simply compare the ratio of the peak areas to find our unknown concentration. But keep in mind, this whole thing hinges entirely on your ability to inject that exact identical volume both times. So what if you can't? Let's look at section 5, method 2, the internal standard. Right, so what happens when you're in a situation where you simply cannot guarantee identical injection volumes every single time? Well, the internal standard method is kind of a stroke of genius. Instead of trying to nail perfect volumes in separate runs, we actually add a known quantity of a totally different marker compound, that's our internal standard, directly into the unknown sample itself. Because the unknown analyte and our marker are sitting in the exact same physical injection, any tiny error in the syringe volume affects them both equally. So by looking at the ratio of their peak areas within that one single run, the exact injected volume literally becomes irrelevant. Now, obviously, you can't just grab any random chemical off the shelf to be your internal standard. There are strict laboratory rules here. First, it absolutely must be pure, and it cannot already be present in your original sample. Second, it needs to be well resolved. You definitely don't want its peak overlapping and masking the very components you're trying to measure. Third, its retention time should be pretty close to your target analytes, just so they experience similar conditions in the column. And finally, its concentration really needs to be in the same ballpark as the expected concentration of your unknown. So when do we use which method? Let's break it down. The external standard method is fantastic if you're doing repetitive industry analysis, especially if you've got a chromatograph hooked up to a super precise robotic autosampler that guarantees those identical injection volumes over and over. But if you're doing highly precise manual injections, or maybe dealing with a really messy sample prep where you might lose a little volume along the way, the internal standard method is your absolute best friend, purely because it is completely volume independent. And finally, Section 6, Method 3, Normalization. We use this last technique under a very specific set of circumstances. Normalization is specifically for when every single component in a mixture elutes from the column and gets detected. If we know for a fact we are seeing the whole shebang, we can rely on a very simple rule. The sum of all components has to equal exactly 100%. Once we establish the relative response factors for each component, we can easily calculate their relative percentages based just on their peak areas. It is a really elegant, mathematically beautiful way to profile an entire mixture without ever needing to know the exact injected mass. So we've seen how that simple mx equals kx times ax equation kind of falls apart in the real world, and how scientists use external standards, internal markers, and normalization to find the truth anyway. But it leaves us with a really fascinating thought to wrap up this explainer. If instrument tuning constantly changes the rules of the molecular world, and our chemical measurements rely this heavily on clever relative comparisons, what other absolute measurements in science are actually just brilliant calibration tricks? Thanks so much for hanging out with me today. Keep questioning the data and keep learning.